Hello, my name is Nate. I own a website called MCAT where I release a whole bunch of free MCAT resources. In this video, we'll be looking and talking about fatty acid synthesis and the pentose phosphate pathway. To make your life a little bit easier, I do have notes that I've already written out for you that cover everything on the slides. You can find those at the link in the description. That way, all you have to do is just sit back and listen just to make sure you understand everything that I'm saying. Fatty acid synthesis is going to be answering the question, what if we have excess pyruvate or acetyl CoA? So we're going to be using glycogenesis to store the excess glucose that's around our blood, but we may also have excess pyruvate or acetyl CoA either from the middle of aerobic respiration or from our diet that we don't need anymore because we already have a bunch of ATP. So it sets up this relationship where glycogen is going to be the storage form of glucose, and fatty acids is going to be the storage form of acetyl CoA. Pyruvate as well, because we can really easily turn pyruvate into acetyl CoA using the pyruvate dehydrogenase complex. So basically, all of this remaining acetyl CoA is going to be stored as fatty acids via the process fatty acid synthesis. So that brings up the question, what is fat? So fats, they're also called triacylglycerols or triglycerides, TAGs for short. They're all the same thing. Basically, we're going to be storing fatty acids as triglycerides, which is what we call a fat. And these fatty acids are basically just hydrocarbon chains, meaning they're just made up of hydrogen and carbons for the most part, that are going to carry energy in their reduced bonds. And these fatty acids are the storage form of acetyl CoA. Remember that acetyl CoA is a 2-carbon unit. Basically, in a fatty acid, we're going to be progressively adding 2 carbons at a time, each of those 2 carbons being another acetyl CoA until we have formed our fatty acid. So glycogen is mainly used for short-term storage. We'll talk about that here in a second. Fats are really good for long-term storage. We're talking about longer than roughly 24 hours at this point. There's a few reasons for that. The main one is because they're really compact. Notice in the picture that just showed up, these fatty acids can stack on top of each other really easily because they do have this very simple hydrocarbon structure. Because of this, we say that they're really good at packing. Basically just the efficiency of which we can stack fats right on top of each other. So just to actually show you the structure of a fat, we're going to be taking these 3 fatty acids. We're going to combine them with a glycerol, a simple 3-carbon sugar, and in doing so, we form a triacylglycerol. You'll notice that all of the fatty acids in the triglyceride don't have to be the same thing. The top one here, this is called a saturated fatty acid because it has no double bonds. It has the maximal number of hydrogens. This one is a mono-unsaturated fatty acid. This one is a tri-unsaturated fatty acid. But long story short, for our purposes on the MCAT, fats and fatty acids are the storage form of acetyl CoA. Because they form these really neatly packing structures, they can store more energy without taking up as much space, as much volume as our other storage molecules like glycogen. Another reason for this is fats are really hydrophobic, meaning they hate water. So whenever they're stored, they're not going to drag any water with them. Water takes up a whole bunch of space. Whenever it's stored in the body, since fats are hydrophobic, they don't have to worry about that. That's another good reason that fats are really good at packing. Additionally, fats are really good for long-term storage because they have a really high energy density. In fact, they are the most efficient ATP producers per molecule on the MCAT. So one fatty acid or one fat is going to produce more ATP than one glucose, than one acetyl CoA, than one protein, for example. They are extremely efficient producers of ATP. So as we start to advance into long-term starvation, they're going to be really good at providing the most bang for our buck. Glycogen, on the other hand, is better suited for short-term storage, meaning it's going to stick around in the body for less than a day, just as our body starts using glucose. But the reason that we use glycogen for short-term storage mainly revolves around speed. The building of fatty acids and breakdown of fatty acids as well takes a long time. So even though fats are really efficient, it's a much slower process, which is why it's better suited for long-term storage, much more so as an emergency reserve rather than a quick way to get ATP. I don't know if you've ever worked out on an empty stomach before, but it's really difficult. You feel really lethargic, really tired. If you've been starving for long enough, one of the main reasons that your workouts on an empty stomach are really hard is because your body's going to start to switch from breaking down glycogen to fats. And since the process of breaking down fats is relatively slow, you're going to feel really lethargic in your workout because your body is not producing ATP at a very high rate. So glycogen is much quicker to break down because pretty much all we have to do is just slice glucose monomers off of that polymer chain, that polysaccharide chain, and then glucose is immediately ready for any type of respiration where we can produce ATP. Glycogen also requires a lot of water to be stored. Sugars in general are very polar molecules, meaning they're going to attract water. So this also kind of explains why it's not really great for long-term storage. It's much more suited for short-term storage when we can easily get rid of it. So now that we've talked about why our body uses and builds fats, I'm going to start by giving you an overview of fatty acid synthesis. We're going to use this slide to basically talk about how fatty acid synthesis works. Luckily on the MCAT, there's not a ton of steps in fatty acid synthesis you need to know. You basically just need to understand the main idea as well as some of the important enzymes involved. We'll cover everything on this slide. So fatty acid synthesis is mainly going to be taking place in the cytosol or cytoplasm of the liver. We specifically call the liver cells hepatocytes and adipose tissue as well. This is just our fat storing cells. So we're mainly just going to focus on the liver for the sake of simplicity. Just know that this is also going on in adipose tissue as well. But in the liver, most of our acetyl-CoA and pyruvate that we're wanting to store in this condition is going to be found in the mitochondrial matrix. So notice that fatty acid synthesis occurs in the cytosol. So we need to move acetyl-CoA back into the cytosol in order to actually store it as fatty acids. This is done by an enzyme called ATP citrate lyase, which essentially for our purposes is just going to throw acetyl-CoA, that 2-carbon unit, back into the cytosol. So ATP citrate lyase is specifically going to be doing this in these postprandial conditions, specifically when we have plenty of ATP and plenty of acetyl-CoA that we no longer need to make ATP. Conversely, if we did need to keep making ATP, we could also just send this acetyl-CoA through the Krebs cycle. But for right now, with what we're talking about with fatty acid synthesis, we're wanting to store this acetyl-CoA, so we're going to move it to the cytosol. Again, that's done by ATP citrate lyase. And we're going to be storing this excess acetyl-CoA, and technically pyruvate, we'll make a comment about that in a second, as fatty acids, our long-term sort of emergency storage. We largely really don't want to be messing with our fat levels that much on a daily basis. Your glycogen levels are going to vary dramatically throughout the course of a day. Fats are much more so a break glass in case of emergency type situation. So the basic idea of what's going on is we're going to be taking acetyl-CoA or pyruvate, which we just immediately turn into acetyl-CoA, and we're going to be turning this acetyl-CoA into what's called malonyl-CoA. This is done by an enzyme called acetyl-CoA carboxylase. A carboxylase is an enzyme that's adding a carboxyl group. Basically, it's adding a carbon dioxide to it. Notice that we jumped from 2 carbons to 3 carbons in this step. Now that we've formed this malonyl-CoA, we're going to use another enzyme called fatty acid synthase to add on an acetyl-CoA, basically. Specifically, it's in this form that we call acetyl-ACP. You don't really need to worry about that too much. And in doing so, we're actually going to re-kick off another carbon dioxide in the process. Kind of weird. We add a carbon in the first step just to remove it a little bit later on. Again, for the MCAT, simplicity is going to be your best friend here. Basically, we are repetitively adding acetyl-CoA units onto themselves. We just have to go through this malonyl-CoA intermediate step. So this process is going to repeat until we form what's called palmitate or palmitic acid. They're the same thing. This is a 16-carbon saturated fatty acid. So we're going to repetitively add these 2-carbon acetyl-CoAs until we get to that 16-carbon number. On the MCAT, they just want you to know that that's when fatty acid synthesis is going to stop, at least within humans. So that's basically how fatty acid synthesis works. I'm going to bring up the important enzymes here again a little bit later. But another point that I've already alluded to several times is that we can either start with pyruvate or acetyl-CoA. Both of them are in the mitochondrial matrix. And pyruvate can just immediately be sent through the PDC to be turned into acetyl-CoA, which is why I've been saying that you can kind of also think of fatty acids as the storage form of pyruvate as well. Another important thing to note is that this uses ATP, specifically the enzyme fatty acid synthase uses ATP. We're going to cover how much specifically ATP is used on the next slide. Again, to reiterate, we are doing this when we have high amounts of acetyl-CoA or pyruvate or citrate, any of those intermediates for those mitochondrial metabolic processes. We've got a ton of those, but we also have a ton of ATP, and we don't need to be producing any more of it. We're going to convert everything into acetyl-CoA, use ATP citrate lyase to send it into the cytosol, and run fatty acid synthesis. Again, as a reminder, this is still occurring within the postprandial metabolic state, where our initial goal is to use our diet to produce as much ATP as possible. And once we've done that, we're going to store everything that's left over. So just like everything else in the postprandial metabolic state, the hormone that's going to be mainly promoting fatty acid synthesis is going to be insulin, which also means that the hormone that will be inhibiting it will be glucagon. There's a few enzymes that you just need to know the basics of. The first is pyruvate dehydrogenase, just because that can convert pyruvate into acetyl-CoA, and then we can send that acetyl-CoA through fatty acid synthesis. The next is that acetyl-CoA carboxylase. It is adding that carbon dioxide onto acetyl-CoA to form malonyl-CoA. This is also considered the rate-limiting step of fatty acid synthesis, meaning that if we've formed this malonyl-CoA, that molecule is destined to become part of a fatty acid. We are no longer going to be using that molecule for anything else. That's what we mean by rate-limiting step, the committed step as it's also called. The main functional enzyme that's actually going to be doing these two carbon additions is called fatty acid synthase. That basically it is just repetitively adding 2 carbon units onto this growing fatty acid. It is the enzyme that's mainly responsible for fatty acid synthesis, which should make sense. It's literally called fatty acid synthase. One last important note is that this enzyme, fatty acid synthase, is going to utilize an electron carrier called NADPH. Slightly different than NADH, and it's going to have a very different functionality than NADH. We'll talk about that later. So that's fatty acid synthesis, the basic steps of it. We're not done yet. Sorry if you're getting excited. We still have a few more things that we need to talk about. But again, long story short, we're shipping extra acetyl-CoA into the cytosol. We're going to turn it into malonyl-CoA and then keep adding on those 2 carbon units while we're doing that decarboxylation via fatty acid synthase, just repetitively adding 2 carbon units onto themselves to form palmitate, that 16 carbon saturated fatty acid, which will serve as the main acetyl-CoA storage for humans throughout our body. We can then take this palmitate, package it up into a fat, and store it for whenever the body needs it. Another thing the MCAT might want you to know, sorry if you can hear cars or motorcycles in the background. I've tried to limit that noise as much as possible, but I live on a pretty busy street, and sometimes there are people who are trying to show off how fast their car is. It's annoying enough that I'm actually moving soon. Not important. The MCAT sometimes wants you to know how much ATP or NADPH is required for the synthesis of a certain fatty acid. So again, it requires both. It's going to require both ATP and NADPH in order to form these big fatty acids that we're storing in fats. So there's a pretty simple equation that's going to allow us to figure out how much of each that we need. What you're going to do is you're going to take the number of carbons that the fatty acid has, and then you're going to set that equal to 2n plus 2. This n that you're going to be solving for is the number of ATP, and you just multiply n by 2 in order to get the number of NADPH. Basically, there's going to be twice as much NADPH used as ATP. So for example, let's take that palmitic acid that we just talked about on the last slide. It has 16 carbons, and so 16 is equal to 2n plus 2. If you solve for n, you're going to get 7. So therefore, the synthesis of palmitic acid requires 7 ATP and 14 NADPH because it requires twice as much NADPH as it does ATP. So you might be asking, what specifically is this molecule NADPH? We haven't seen it anywhere else in metabolism so far. So what in the world is this thing? Where did it come from? Where did it go? Where did it come from? Cotton Eye Joe, I guess, whatever. So NADPH is simply going to be NADH with a phosphate attached. So it's going to have a very similar nucleic acid-based structure as NADH, but it's going to have a dramatically different function. They look similar, but they're not going to behave similar. NADH is specifically going to be used to make ATP at the electron transport chain. That's its job. NADPH, however, is going to be used for 2 different main things. The first is to protect the cell against what we call reactive oxygen species, ROS for short. These are just really reactive forms of oxygen that are just formed in normal metabolic processes, as well as a lot of our immune reactions, things of that sort. Basically, sometimes on accident, our body's going to produce these ROS that we need to just take care of. And NADPH is basically going to sacrifice itself in order to do so. So it protects us against ROS. It's also going to be used in the redox reactions that we're doing in fatty acid synthesis. So NADH and FADH2, we saw those in all of our redox reactions in the rest of metabolism. Specifically in fatty acid synthesis, we're going to see NADPH instead. So you might be asking why? Why would we need a separate electron carrier? The first important reason is the location. Remember that fatty acid synthesis is occurring in the cytosol, whereas most of our NADH and FADH2 is largely going to be produced inside the matrix. They're largely going to be residing within the matrix. So it would be kind of annoying for us to have to constantly ship NADH or FADH2 back and forth from the matrix into the cytosol. I'm going to fix this FADH2. The 2 is in a subscript, and that's really bothering me. Give me a second. Okay, my OCD is resolved. So basically, because we have this separate electron carrier, that its job is actually to reside in the cytosol, that allows us to have a separation of these two processes, where basically the electron carriers that stick around in the mitochondrial matrix, NADH and FADH2, they only have one job, which is basically to produce ATP. And then NADPH, which is going to hang out in the cytosol, it's going to have its jobs that are completely different. So therefore, how do we make NADPH? Through a process called the Pentose phosphate pathway. This is going to be another process that's going to be occurring in the cytosol. So the Pentose phosphate pathway, or PPP for short, it is going to be in the cytosol of pretty much every single cell. It actually has a few different goals that go beyond producing NADPH for fatty acid synthesis. Basically, every single cell is going to need NADPH. We're actually going to produce nucleotide precursors in this process as well. So because of that, the PPP is not limited to the hepatocytes or adipose tissue. Those two types of tissue are specifically going to be taking advantage of that NADPH to make fatty acids, but they're also going to be doing several other things with it as well. That's what we're here to talk about. So the goal of the PPP is to divert glycolytic intermediates, certain molecules from the middle of glycolysis. We're going to shunt them away from glycolysis for a short bit, and we're going to use them to produce NADPH or potentially nucleotide precursors if they're needed. We're going to talk about this in a second. We also call this the G6P shunt because basically we are temporarily removing G6P, glucose 6-phosphate, from glycolysis. We're going to run some reactions with it to produce all of this stuff that we need through the PPP, and then we're later going to return it back into glycolysis. There's two phases of the PPP that you need to know about. We're going to talk about these way more in depth on the next slide. But basically, the first phase is called the oxidative phase. It's going to be catalyzing a redox reaction. So, of course, that's the phase that's going to be producing NADPH. That's the main goal of the oxidative phase. It's also important to note that this phase is irreversible. The non-oxidative phase, meaning there's no oxidation going on, it is a reversible phase that has the main goal of either producing nucleotide precursors if they're needed, or if they're not, we're basically just taking the product of the oxidative phase and returning it back into glycolysis. So because this is the main pathway that is producing both NADPH and nucleotide precursors, this sets up two main scenarios that we're going to need the pentose phosphate pathway. The first is, of course, if we need NADPH, because NADPH is used to form fatty acids if this is a liver or a fat cell, but also all cells are going to need protection from ROS as well. So that's the first reason that we'll run the pentose phosphate pathway. The second, of course, is whenever we need nucleotides. So mainly during the synthesis phase of the cell cycle, the S phase, we're producing a whole bunch of DNA when synthesizing that complementary strand of DNA. So because we're essentially building a brand new strand of DNA, we're going to need a whole bunch of nucleotides. We're going to need a whole bunch of ribose 5-phosphate, which is going to be the building block of that deoxyribose sugar that we're going to be using in DNA. So because this is the phase that we're producing a whole bunch of these nucleotide precursors, the PPP is going to be really active during the synthesis phase of the cell cycle. And of course, we're still within the postprandial state here. So as you can probably guess, going to be promoted by insulin. So to explain how the pentose phosphate pathway works, we're going to start with, of course, with forming that G6P that we need. This is just done by hexokinase or glucokinase, depending on what type of cell we're in, of course. But once we've formed this G6P, we're going to divert it from the rest of glycolysis by first sending it through the oxidative phase. Again, as a reminder, this phase is irreversible, meaning that it will only go in one direction. Notice that this arrow is only pointing away from G6P. Within the oxidative phase, we're going to be taking G6P, and through a series of steps that you don't really need to worry too much about, we're going to be oxidizing it into the 5-carbon molecule ribulose 5-phosphate. And since we've lost a carbon, of course, that's going to be lost as carbon dioxide. So there's a decarboxylation happening here. This is also an oxidation of G6P, which will allow us to reduce NADP into NADPH. Specifically, for every single G6P, we're going to be able to produce 2 of these NADPH. This is largely done by the enzyme G6P dehydrogenase. Remember that dehydrogenases do all of those redox reactions in metabolism. And because it's irreversible, that means it's going to have some sort of regulation that you need to know about. Specifically, G6P dehydrogenase is inhibited through negative feedback by high amounts of NADPH. If we have high amounts of NADPH, we're going to shut off the oxidative phase. There is another enzyme that helps us with the production of ribulose 5-phosphate. It's called 6-phosphogluconate dehydrogenase. You don't need to worry about it for the purpose of the MCAT. In fact, you don't need to worry about any other enzyme for the pentose phosphate pathway outside of G6P dehydrogenase. So we're going to be doing the oxidative phase whenever we need NADPH, because that's the main goal of the oxidative phase. The next phase, the non-oxidative phase, is going to be reversible. So you're going to see some two-way arrows here in a second, where we're going to be taking ribulose 5-phosphate and turning it into ribose 5-phosphate. Of course, like we've talked about, that word ribose should sound familiar for the purposes of nucleotides. So if we need nucleotides, we can just stop the reaction here, take this R5P, and start using it to build nucleotides, which we're going to need for DNA synthesis. If we don't need nucleotides, we're basically going to start sending this through some other reactions to turn it into F6P or G3P. So because they're glycolytic intermediates, if we don't need these nucleotides, we can basically just return them straight into the rest of glycolysis. So basically, within the non-oxidative phase, we're either going to be returning the carbon chain back to glycolysis, or we're going to be leaving it as R5P in order to build nucleotides. So we talked about on the last slide the fact that we need the pentose phosphate pathway for those 2 reasons of producing NADPH or nucleotide precursors. There's actually going to be 3 different scenarios or orders in which we're running the pentose phosphate pathway. For example, what about the scenario that we only need NADPH? We don't need nucleotide precursors, we only need NADPH. In this scenario, we're basically just going to run the oxidative phase straight through, which of course is going to produce that NADPH that we might need for whatever reason, whether it's fatty acid synthesis or protection against ROS. And then we're basically just going to utilize the non-oxidative phase with the goal of forming F6P or G3P, those intermediates that can just go straight back into glycolysis. If we need NADPH and nucleotides, we need both, we're still going to run the oxidative phase in order to produce that NADPH. And then we're going to stop the non-oxidative phase right after we've made that ribose 5-phosphate. Finally, the last scenario is whenever we only need nucleotides, such as the synthesis phase of the cell cycle. If we only need nucleotides, we are actually going to be running the non-oxidative phase in reverse. We don't need NADPH. We don't need to be running the oxidative phase. So we actually have a way of shuttling F6P and G3P backwards through the non-oxidative phase of the PPP in order to produce R5P. It's also important that this phase is reversible. The non-oxidative phase is reversible because, like we talked about earlier, NADPH inhibits the oxidative phase. If we have high amounts of NADPH, eventually that will start to shut off the oxidative phase, which will stop the production of ribulose 5-phosphate and eventually ribose 5-phosphate. So we need to have another way of forming R5P in order to make nucleotides. That's why the non-oxidative phase is reversible. That's why we have this second arrow that is going backwards through the non-oxidative phase. Like I've mentioned, the only important enzyme that you need to know for the PPP is G6P dehydrogenase, but understanding these 3 different scenarios is going to make your life a lot easier in understanding when the pentose phosphate pathway is going to be run. And of course, since those enzymes are in blue, it's a process that's promoted by insulin. So that was fatty acid synthesis and the pentose phosphate pathway. We actually are getting pretty close to the end of metabolism here at this point. Most of our metabolic processes that you need to know for the MCAT heavily reside within the postprandial state, and we're getting close to the end of the postprandial state. In fact, all we have to do is just summarize it now at this point. But just as a reminder, if you need any of my other free resources, you want to go back and watch any other videos, you want to find any of my notes, anything like that, if you want to suggest some sort of free resource that you need help with that you want me to make, you can find all of that on my website. That's linked in the description.